Morning. I am here to introduce the second speaker of the day, who is Dr. Damaris Odeny, who works for the ICRISAT. So first of all, my name is Mesifa Gude. I am a third-year student in plant breeding in Dr. Thomas Lubastad lab, and I am happy to be here today. So Dr. Odeny is joining us from India. No, it's no longer Kenya. I thought she was gonna be in Kenya, but she just let me know that she's now in India. So we appreciate her coming to give us this talk. So Dr. Odeny is a plant breeder with research experience in Africa, Asia, Europe, and US. She has led and implemented genomics projects in both cereals and legumes, including the development of genomic resources in several orphan crops. She works closely with other disciplines from national, private, and international institutes to develop cutting-edge genomic resources for all the ICRISAT mandate crops in her current global role. Prior to her position in ICRISAT, she worked as a senior researcher at the Agricultural Research Council in South Africa, where she played a key role in establishing molecular breeding processes for indigenous vegetables. Dr. Odeny is also passionate about mentoring young upcoming scientists and has successfully mentored about 50 scientists in her career to date. This morning, Dr. Odeny will be presenting developing genomic resources for climate resilient crops. Please join me in welcoming Dr. Odeny.
Hello, everybody. I don't know if my video is visible. Can you hear me?
Yes, we can hear you.
Okay. Thank you very much for the opportunity to present at this forum. This is my first time interacting this close with Iowa State. It is a great opportunity for me, and thank you so much. And thank you also, Marcy, for that wonderful introduction. So I'm going to go straight into my presentation. Just a brief outline. Okay. Yeah, so I'll be talking about sorghum bicolor, finger millet, which is Elusine coracana, and groundnut, which is also referred to as peanut in the US. This is Arachis hypogea. And I'll be talking from molecular marker development all the way to trait validation and integration. And also just to say from the beginning that the work that I'm presenting here is undertaken through capacity building. So every activity that I do, I incorporate training a postgraduate student, an intern, and so forth. And I just want to thank in advance my collaborators, the students, the fellows that I've worked with, and also colleagues who've contributed to the work that I'm going to present. I'll start with the marker development work that we've done on finger millet, and then I'll shift to the other crops as well. And for those of you who haven't, who've never seen finger millet, it's a tetraploid. It's a crop that is native to the African continent. Considered probably one of the most, the oldest domesticated cereals. It's thought to have been domesticated around the highlands of Ethiopia or Uganda, so somewhere around here. And you can see that also follows the regions where it is most popularly grown in Africa. It's also grown in India, so it must have been introduced into India during the trade route. So it's also grown a lot in the southern parts of India. But finger millet, the popularity is really increasing, and now you find it grown, the cultivation is actually spreading quite a bit also in different parts of the world. Now, this wonderful crop is highly nutritious. It is resilient to a lot of stresses. It is actually one of the most calcium abundant cereals. So the calcium content is up to 10 times more than you'd find in wheat or maize or rice. Now, despite being that wonderful, it is very little research. So when I started working on finger millet about 10 years ago, there were only 20 simple sequence repeat markers that were available, and they didn't always work because it's a tetraploid, and some of the markers were not genome specific, so you got mixed signals. So one of the main challenges that we really wanted to address was the fact that a parasitic weed called Striga hamontica, which you can see here on the screen, was getting such a big problem. But then it was really difficult to even do any genomic selection, genomic analysis on it because there were no resources. So we started by developing or doing whole genome sequencing. We did this work in collaboration with the BGI, Beijing Genomics Institute. We did short read sequencing at the time. That was like 9 years ago. And yeah, short reads were the in thing. And we prepared libraries from 250 base pairs up to 20K. The larger libraries were mate pair libraries. So we used the short reads and were able to estimate the genome size to about 1.45 GB, which also corresponded with the flow cytometry estimates that were done before. And we did a pretty good job with the short reads to generate a good representative reference genome, as you can see on the screen. If you're interested in the statistics, you can see that on the screen. But that made us to be able to start more or less like open a new era for finger millet sequencing of genomics. We were able to do whole genome marker discovery. We were able to do genotyping by sequencing and call SNPs more accurately. We were able to do linkage mapping and trait discovery and characterization, like doing transcriptome analysis. The next thing that we ended up doing, and I think this is also one of the things that a lot of genomic scientists really struggle with, is you end up generating all these amounts of data that you're not able to use regularly, because in your routine breeding program, you just need a few markers that work well. Those are the ones you concentrate on. And so the next chapter we got into is to now move into developing a few very high informative markers, not just for finger millet, for the other crops as well. And as you can see from the screen, yes, so we used the whole genome sequences that we generated, plus the SNP markers that we generated, and developed QC markers. Now, to be specific, most of the QC markers you see developed here were developed by other groups, and what we did was just validate the markers using the breeding lines from the region. And these breeding lines are not just from ICRISAT, but also from the national programs, because one of the things that we do as ICRISAT is to support the national partners as well. And so in the case of finger millet and sorghum, the markers that were developed here were already existing and were developed by our colleagues from ICRISAT headquarters in India. And so we went ahead and validated them. And as you can see, we were able to validate 15 for finger millet, 30 for sorghum. The good thing is they are evenly spread across the genome, so they do a pretty good job. In the case of groundnut, we did a lot more, because beyond the 20 that was already existing, we also developed about 60 more by ourselves. And then we also got a little more that were developed by CERAS, which is an institute in Senegal. And the additional ones were developed by Daniel Fonseca. And we were able to validate 106 of them. Now, with groundnut, because we had a larger panel of QC markers, we were able to come up with a really good set of markers. And if you can see here, I think one of the most important characteristics of QC markers is that they should be able to, the top 10 or the top few markers should be able to explain a high genetic variation amongst your breeding lines, so that every time you are trying to detect any contamination, or you're looking to look for successful hybridization, you only need to test a few. And you can see with Groundnut, we've been able to do a pretty good job. The top 10 markers out of that set are explaining up to 64% genetic variation across the first two PCs. Now, we still haven't gotten there with finger millet and sorghum, but we are on it. Sorghum, we have the first two PCs explaining about 45% genetic variation, and we really want to improve it. I think the level where we want to be is to be able to have the first top 10 or at the most top 15 explaining more than 80% genetic variation across the breeding lines in the region. And the breeders in the region actually do share a lot of germplasm or breeding lines. And so this is quite possible to achieve. And you can see here at the bottom, I'm just giving an example. The current first top 10 QC markers we have in sorghum, we tested them recently in a breeding program where these two parents were used to generate F1s. And as you can see, out of those 10, 3 did a pretty good job and were quite consistent in confirming successful hybridization of these progenies. So this is pretty good. I mean, you only need 1 or 2 that work well, and if you have 3 or more, it's a bonus. So these are already practically being used in the breeding programs in Eastern and Southern Africa. So one of the other things also that is becoming very common to do in most breeding programs is the development of mid-density marker panel. And this is especially for crops where, you know, a lot of trait marker discovery has happened, and there are a set of markers that are known to work or are associated with a certain trait. So then you just need a good representation of those markers, and you can use it for genomic selection, genomic prediction, or QTL mapping, or you know, to define heterotic groups, as all the breeding applications where you need a little bit more markers than the QC markers can do. And so we've done that for the 3 crops as well. You can see on the screen the numbers that we have and the ones that we have validated. Now, because we are still very much on in the trait discovery stage for most of these crops, we are not yet where we are using these for genomic selection. And actually what we need to still do is, you know, do a lot more trait discovery and validation, and then incorporate most of the markers that we're using into these panels so that in future we can actually use them for genomic prediction. But, you know, as a test, we just wanted to check, you know, how informative are these markers. And you can see on the screen, I've provided the ones for sorghum and finger millet, and they're able to differentiate the Eastern and Southern African material pretty well, both for sorghum and finger millet. So I'll switch a little bit and go into trait discovery. And the first I'll start with is sorghum. And the trait I'm going to talk about here is biological nitrification inhibition, which some of you may know this trait, some of you may not. But I've been so fascinated by this trait because it is the ability of the crop to be able to inhibit the nitrification process that normally leads to the leaching of nitrogen or converting nitrogen in a state that cannot be uptaken by the plant. And that is actually one of the best ways to contaminate underground water. And also this is how you end up with a lot of greenhouse gases. Now, this trait has been of great interest to me because it is applicable both in high intensification production systems, but also in smallholder systems. High intensification system because people tend to just use nitrogen all the time, and then it gets wasted. And they do that because most of the time after you apply nitrogen in whatever form, in ammonia, it gets converted into something else. So you can just apply one season and the next season there's not enough for the plant. But in smallholder systems, it's also the case that most farmers cannot afford to apply nitrogen. In fact, most of them actually just depend on the intercrop or the nitrogen that is fixed by the legumes that they intercrop with. And so whatever it is that is fixed, they need to have it available as much as possible. Now, in sorghum, a compound called sorghum has been identified as the compound that helps with the BNI, with the biological nitrification inhibition. And the pathway for sorghum synthesis has been well characterized, well published. If you want to know more, you can actually go to that publication and have a look. But since this was such an interesting trait, especially very relevant for climate resilience, we wanted to look at, what are the genes associated with the production, or what are the markers that we can use to understand the genetics and how we can introgress this trait into farmer preferred varieties. So we started by looking at two kinds of populations. We looked at a diverse set of population, and especially because most of it is still not characterized. So most of the genotypes we have, we do not know whether they contain the trait or not. So we used a subset of the sorghum reference set. Those of you who are working in sorghum, working on sorghum may be aware of the reference set. So we used a subset of 231 and phenotyped them. This work has been done in collaboration with colleagues at JIRCAS in Japan. In fact, colleagues at JIRCAS, actually, Dr. Subarao, championed the work on BNI in other crops as well. So they do the phenotyping. The phenotyping is quite labor intensive. It takes a long time. In fact, it took them a whole year to phenotype the 231. But we also did develop a recombinant inbred line mapping population between these parents, in which PVK801 is a high producer sorghum and 296B is a low producer. So we had segregating population, and both of these populations were phenotyped at JIRCAS in Japan. So, and I forgot to say that the genotypic data for both these populations we generated using genotyping by sequencing. So basically just low coverage sequencing at 0.5x, and then imputing, and then filtering, and using the SNPs that are retained. Now, looking at the phenotypic data that was generated from the phenotyping that was done in JIRCAS was pretty good. We have a good variation across the genotypes that we used. You can see that the genotypes also showed significant variation among them. So, and the heritability was pretty good. So we were good to go with GWAS analysis, and this was from the reference set. When we did GWAS analysis, we were able to pick up some interesting peaks, and I'm just going to give two examples here for the root dry weight and sorghum content. Just to say that, the root dry weight and all these other traits that we measured, it's basically to try and let us know, how much sorghum is produced in one versus the other. It's just that, in sorghum content, you're looking at the micrograms of sorghum per 0.5 ml, or if it's SSC, you're looking for it in different measures. But at the end of the day, you're basically measuring the same thing. So yeah, we were able to find a nice peak on chromosome 8, explaining some kind of association with the root dry weight, and we looked at the candidate genes, but we were also able to find an interesting QTL on chromosome 4, also with interesting candidate genes, explaining or showing association with sorghum concentration. Now, we were hoping that when we mapped the recombinant inbred line mapping population, that we would be able to find some overlap between the QTL from the GWAS, but yeah, we still got really good results. As you can see, we have significant variation across the real families, and a very good variation for the trait, the different traits measured. But in this case, we were able to find the peaks on QTLs on chromosome 7, and results you're seeing on the screen there are both on chromosome 7, using both ICIM and QTL cartographer. So we looked at the one that was consistent, which is specific Sorghum, was consistent across the different methods, and found this particular locus on chromosome 7. And when we looked a little bit closer, we were able to see a cluster of MIB family transcription factors, which we know are involved in signaling. But we were also quite surprised to see a synthase-like family, bearing in mind that, when I showed the pathway earlier, there's a synthase that is involved in the synthesis. So these were quite interesting traits for us. And we are looking at them a little bit more keenly and doing a lot more characterization. And something else that we've been able to do is that we found, so the biparental mapping population we used here had a good enough variation, contrast for Sorghum production. But then afterwards, we screened a little bit more genotypes that were from West African pool, and we've been able to find even more higher Sorghum producers, and we've been able to generate mapping populations, and we are phenotyping and genotyping them at the moment. So I'll switch from sorghum and then go into groundnut. Just talk about a little bit about groundnut rosette disease. Now, this disease is the most devastating peanut disease in Africa. It was reported over a century ago, and not much has been done, which is a little bit embarrassing. And as you can see, all these states highlighted here, green, have challenges with groundnut rosette disease. Now, the work I'm presenting here was actually done by Esther Achola, who is a PhD student at Makerere University. And she had more objectives, but what I'm going to highlight here is just the work that we did in terms of identifying genomic regions associated with GRD. And also just to say that this paper, her paper, just got published recently. If you're interested, you can have a look into more detail on what we did. And so, I just forgot to mention that this is how GRD looks in the field. You will not have seen it in the US because it only exists in Africa, but it is pretty devastating. So you find all the obvious virus symptoms: yellowing, curling of leaves, and also, okay. I don't know if I'm supposed to do something. Expand window to show video. Maybe it's not meant for me. Okay. So Esther did quite a bit of work. She tested an African mini corset. So what the breeders did was they collected breeding lines from all these countries that are highlighted here, and they collected the breeding lines, genotyped them, and came up with an African mini corset. Now, that is the set that Esther was able to phenotype across 2 locations in 3 seasons in Uganda, in Serere and in Nakabango. Now, unfortunately, the disease pressure in Serere was not that high, so we ended up not getting good heritability for the trait at that location. But for Nakabango was pretty good, so we decided to proceed with the Nakabango data set for GWAS analysis. The markers that we used here were generated, were from an Affymetrix chip that is developed from the progenitors of Arachis apoge, that is Hypaeasis and Duranensis. But the markers were really informative. As you can see, these markers explained more than 60% genetic variation across the 3 PCs. Now, the results were really good. We were able to identify 2 consistent peaks on chromosome 4 and chromosome 14. Now, for those who are working on groundnut, you know that chromosome 4 and chromosome 14 are more or less synthetic. So this was quite exciting for us to see the consistency here. We also had an additional QTL here, but it wasn't as consistent as these two. So we focused on these two QTLs, also looked for additional, if we could detect some obvious haplotypes, which we did, and identified these 5 haplotypes. And one of them was actually specific to Ugandan collections, which was not surprising. We've also reported in that paper, you know, the additional haplotype work that we tested. We went further to look at candidate genes, and it was quite interesting. I mean, this is one of the few GWAS analyses that I've done where out of, I think we identified about 34 markers that were associated with the trait, but half of those were sitting right within genes. So half of them were actually functional markers. And you can see all the interesting genes of interest that we were able to pick out. And I'm just highlighting here, you know, the fact that we were just able to identify, you know, NBS LRR resistant gene, also just having 2 markers sitting on it, and Argonaut family protein, especially in this case, we have virus that we are working with. And finding these 2 is definitely not a coincidence. You know, we know that antiviral RNA silencing involves both NBS LRR proteins and Argonaut protein. And so what we're looking at now is, you know, are we able to associate any short RNAs or small RNAs with this kind of resistance? Are we able to see some clear patterns in terms of what happens in the resistant lines versus susceptible lines? But beyond that, also we're doing marker development to be able to come up with CASP assays that we can routinely use for routine selection for resistance to GRD. Well, granted, this was just the beginning, and it seemed to have worked really well thanks to the efforts that Esther put into doing good phenotyping. But we still do need to generate more mapping populations, validate, and do a lot more validating to be able to select with confidence. But yeah, I mean, we are really going to the right direction because most of these markers explained at least 40% of the phenotypic variation that we observed. So that worked really well. So I'll shift gear a little bit and then go into just some examples of trait marker development and validation. And here I'm going to talk about striga resistance, drought tolerance, and just a little bit on fertility restoration. So we got, these were already existing markers that we decided to test and just to make something available quickly. And we tested 8 markers for striga. In this case, we only had 100 genotypes that had historical data on their response to striga. And when we tested, we didn't get really very good reproducibility in terms of, you know, it was only 31% of the time, and this was the best scenario, only 31% of the time where we had striga tolerance, we could see some of these alleles. So this didn't work very well because 31 is a little bit low for selection. And so we'll need to do a little bit more. But this was not surprising because striga is such a highly evolving pathogen or parasite. And then the mechanisms of striga resistance are also not that well known. You know, some are pre-attachment, some are post-attachment, some are mechanical. It involves a lot more things. And then there's also the environment factor that plays a completely different role. So this just tells us that we need to understand striga a little bit more, but also develop markers that are more reliable for selection of, hopefully, all the kinds of variations that we are able to see in the field. So that didn't work very well, but I have another good example that worked really well. So these are stay green markers that have already been published. They are available. They are known by most people. And what we did was just, you know, take the already existing markers and try to validate them across drought-tolerant breeding materials that are available in the region. And this region, well, every time I mention region, I mean Eastern and Southern Africa. And it worked really, really well. We used historical data, and whenever we had genotypes with historical data showing that they're consistently drought tolerant, they always 100% of the time had favorable alleles of these 2 markers at the top and almost 99% times the next two, and more than 80% time the following two. And so these are now available for deployment right away for drought tolerance screening, for breeders who are interested. And the good thing with these markers is the breeders don't need to have these working in their situation. They just need to sample their plants and submit to Intertech. So basically to say that this whole marker development is done in collaboration with Excellence in Breeding platform. And I think what they have done is really, really great, because then it's giving access to most of the breeders in the region who do not even have labs to be able to utilize the tools, the basic tools for identification of markers in their programs and introgression. It would be good to see how many of the breeders are actually using it. But for those that are working together with us, that we are providing support, we actually do support them in submitting the data and also making use of the data and translating it in a way that they are able to apply. The last one I'm giving here is fertility restoration, especially for those working on hybrid seed production in sorghum. For these also, we tested 13 markers, and those that worked relatively well were about 3, and especially the first one worked 90% of the time. And this is also something that can be used right away. We did use about 153 maintainers for this study. So in summary, I'm just going to wrap up and say, there are exciting opportunities for climate resilient crops. There are many traits that remain uncharacterized and unknown. There's just so much that needs to be done. And there's a lot of learning from both well-characterized or well-studied crops like maize, rice, wheat. But I think those crops also have a lot to learn from these underutilized crops. So there's a whole new window there for learning to be done across the different crops, but also across different institutions. As I mentioned already, I'm very passionate about capacity building. This is a great opportunity for me to also just do an open call. If there are any students from Iowa State who are interested in spending some time in Africa and have an experience of really seeing the challenges that the smallholder farmers are tackling, you're welcome to come to my lab and spend some time there. Enjoy seeing the countryside and the beauty of the country, and enjoy science as well. And just to finalize by probably going to embarrass Anne now, but Anne was actually a very significant part of the lab before she joined Iowa State as a PhD student. And, I've always had quite a number of young upcoming women scientists in the group, also because I believe that they are underrepresented. But we have men as well. And I'll be more than happy to welcome more in the lab as well. We have different kinds of capacity building, internship, postgraduates, short-term training, theme-specific, all sorts of trainings that go on in our lab. And just to finalize by saying thank you to our donors. We've got support from so many different groups, from so many donors, and also from many partners, private, public, all sorts of partners. And also to thank you for listening. I'll be more than happy to take some questions. Thank you.
Dr. Odeny for that wonderful presentation. So we can open the floor for questions.
Thank you for the presentation. Can you actually please elaborate on the Sorghum, and then how did you screening the Sorghum content or the level to conduct the mapping experiment?
Yeah, great question. So you wanted me to elaborate on how we did the phenotyping?
Yes.
Yeah, so actually the phenotyping was not done by our group. As I mentioned, the phenotyping was done by JERCAs. They've been doing the phenotyping over the years, and so they're really good at it. It basically involves setting up the experiment such that you are able to capture or measure or capture the compound that is released by the root hairs, because Sorghum is produced by the root hairs. So they insert, I think, certain tubes in the soil and are able to capture the compound and then do HPLC or some kind of GCMS to be able to quantify the Sorghum that is trapped there. So they do for us all the phenotyping because they've really perfected it. And I know that there are different groups also in the US that are trying to figure out a simpler way of phenotyping. I know that Texas A&M, there's a group that have really been trying to fine tune it. But our part has been mostly, the generation of the genotypic data and the association analysis and also provision of the germplasm.
Thank you.
Hi, Dr. Odeny. Thank you for your presentation. Yeah, I'm just curious about the biological nitrification inhibition. How much G by E have you observed by evaluating your germplasm across different locations? Thank you.
Yeah, so that's an interesting question. Now, if you see the way that we are phenotyping, the phenotyping actually limits you. And this is why we're trying to find more indirect ways of phenotyping for this trait. Because at the moment, we can only do the phenotyping, at least our group can only do the phenotyping with JERCAs in Japan. We haven't been able to venture out to do the phenotyping ourselves across different environments. So that would be something that would be interesting to figure out. But as I mentioned, there are different groups now that are trying to figure out simpler ways so that they know that they can actually look at G by E by evaluating the same set of germplasm across different regions. But as I mentioned, it is quite difficult at the moment, based on what we are using. It's quite difficult to phenotype across environments. As I mentioned, it took a year for them to phenotype the 230 of the reference set.
I have just a quick comment. I was wondering if you were aware that this is 2023 has been declared the International Year of the Millets by the United Nations. So it's a very timely presentation. Thank you for it.
Yes, I'll also just say that, yes, I'm very aware, and ICRISAT is one of the institutions that are involved in the promoting the International Year of the Millets. We just came from India, Delhi, where the Prime Minister actually launched the International Year of Millets Congress. And there will be a series of meetings coming up this year, several in India, but we're also organizing a major one in Nairobi, Kenya around August. So those students who might be interested or who are working on millets, you might want to join us for this occasion.
Thank you. And I do have a question about groundnut breeding. So in the U.S. peanut breeding programs, one of the important issues is allergenicity. And I'm wondering if that's an issue in Africa or really not.
It actually isn't a big problem in Africa. But one thing I do think we are beginning to see is that when you have people who are natively African spending time in the US, or if they are born in the US, they give birth to their offspring in the US, when they come back, they show susceptibility, so they show allergic reactions to peanut. But mostly, it is not a major problem in the region. It is not at all. You find very few cases where there is allergenicity problem. But it could be, it may have to do with the fact that, you know, most African communities actually use groundnut or peanut in the weaning of the children. I think recent studies show that if you are exposed to peanut when you are little, then chances of having allergies to it are very low.
Hi, Dr. Odeny. Wonderful work and a great talk. I have a question related to the slides of post-flowering drought-resistant for validation. Could you please go back to that slide? So I noticed that the, I can't remember how many, it's 9 or 6 SNP you picked for the validation. They are all located at chromosome 2. Could you please explain more why? Yeah, this non-marker.
Yeah. Yeah, so so the stay green trait has been, so there are two, okay, there are a few loci that have been identified to have, yeah, QTL associated with the stay green trait, but the ones that are significant are stay green 3B and stay green 3A, which are the ones you have on the screen here. And so when you're developing, generally when you develop, well, I think from our group, for us to develop markers for integration of a trait of interest, it should explain at least 40% of phenotypic variation. And these two loci, at least with the breeding lines or the mapping populations that we had, were explaining, I think one is explaining about 38, another one about 42%. And so they are relatively major QTLs. And for that reason, it is justified to use them for integration. And especially in this case, you know, we are just not doing genomic selection. We're doing a few QTLs. So we chose to just focus on these two QTLs that are showing higher phenotypic variation that is explained. They are both on chromosome 2, STG3B towards the bottom of chromosome 2 and STG3A as well. These are the two major QTLs for stay green, at least for the lines that we are working with.
So we do not have questions on the chat, so I guess further.
So I have one more question from the chat, actually. Sorry, Mercy, for that. First of all, Dr. Odeny, thank you for your presentation and for dedicating your time for presenting with us. We really appreciate that being in India with such a time difference, you put up with us for presenting and showing us your research. So the question from the chat is there any adoption of push-pull strategies on controlling Striga?
That's a really good question, and I don't know if I'm the right person to answer, or I'm going to. Yeah, I probably need to declare no conflict of interest. So push-pull was developed by ISIPE, which is an institute based in Kenya, and it actually works really well. It's just not sustainable for the farmers for two reasons, and there has been very low adoption of it because the kinds of crops that are part of the complex. So you know you need napier grass, which is the push, and then you need desmodium, which is the pole. And unless the farmers have livestock so they can have napier grass, they are not going to grow it. And desmodium also, they had challenges with obtaining seed, and so most farmers did not adopt it. We've had recent discussions with ICP, and we're trying to use sorghum and alternative legumes. So other than groundnut, ICRISAT is also working on chickpea and pigeon pea. And what we were suggesting was, can we test these other legumes, which farmers will always use, and see if they are able to adopt the push-pull strategy. But at the moment, the adoption is extremely low.
Thank you for that. We have another question from the chat. And regarding to the GRD resistance that you mentioned, that you evaluated materials from that were collected from different breeding lines. So which is the genetic variation that are among breeding programs for selecting those lines in those different places for testing for this resistance?
Okay. Maybe I'll seek to get a little bit more clarification. Is the question asking if there's enough genetic variation for the trait across those breeding lines?
I think it's more regarding the genetic diversity that exists for the different breeding programs across Africa.
Yeah, so I mean, when we look at specific breeding programs, most of the time we don't find sufficient genetic variability for groundnut if you're looking at just one individual breeding program. But when we looked at this diverse set, and that's one of the things that you can actually pick out from our paper that I was mentioning, we get quite good genetic diversity. I didn't include the slide here, but as you can see, even on this slide, you get a pretty good set. It's diverse enough. It's actually pretty diverse. And when we looked back at the genotypes that we found that were showing resistance, we also found them to be quite nicely well spread across the set of germplasm, the African minico set that we use. So it seems to be pretty well representative of what we might need to have as sources of resistance to GRD.
So no further questions, Dr. Odeny. Thank you for taking the time to compile this wonderful presentation, and thank you for joining us today. We appreciate it. Thank you.
Thank you. Thank you too. Thank you everyone for listening. Thank you.